cell. She then went to Tulane, where she trained and was a postdoc with Natalia Troyanova, who many of you know. Natalia and her entire lab spent, if you saw our lab, we divided into half, and half was Natalia and half was us, during the Katrina story. And so there she continued to do work that I'm familiar with on ischemia, but at a larger level, the whole heart, using bi-domain models, if I remember correctly. And then she joined the faculty at Oxford University in the UK, which is currently a professor of medical computing or computational medicine. You can
whatever.
call it that way. But it's in the department, it's based in the Department of Computer Science, which is a nice thing. They're expanding in the direction of computing in biosystems. And she's also a fellow of the Wellcome Trust. So welcome to St. Louis.
Thank you. Thank you. It's very exciting to be here. I mean, I've spent my whole research career admiring Yoram's work and the work that has been done here. So to actually be here, it's amazing. And I've had very good discussions with a lot of faculty today. It's really a privilege to be here. So the first thing is first, I'd like to acknowledge the people who have contributed to this work, and especially people in my group who have done all the research. We managed to get a picture just before I came. I said, I need to show a picture of the lab. And we gathered. Yeah, there are a few people missing, but it's most of them. And very importantly, I would like to acknowledge all the collaborations we have, and our work is purely computational, but we collaborate with a lot of people who provide experimental data, and with whom we have a very long-standing collaboration, not only to build the models, but to test hypotheses and do really the work jointly. So we are very interested for a few years now. We have been very interested in understanding variability and the population heterogeneity, and trying to generate ideas around what makes it similar, what makes a cardiac phenotype normal, and what makes a cardiac phenotype abnormal. And we've done that to understand who may get an arrhythmia, and also to understand mechanisms of arrhythmia. So a lot of the research that is being conducted is to investigate what are the reasons for population heterogeneity from a mutation point of view or a disease point of view. So how does disease lead to a phenotype that is abnormal, or how mutations lead to a phenotype that sometimes is abnormal, but most of the time is normal. So what we have seen also over the years is how environmental factors that are external to the cell, control this heterogeneity. And this is not disease, but it's things like circadian rhythms. So here's a paper that talks about how circadian rhythms modulate cardiac repolarization through changing ionic currents, the QT interval. Food, what we eat, so for example, grapefruit alters very much the response to terfenadine, and this was something that my pharmacologist, my collaborators in pharmacology were telling me, don't drink grapefruit ever if you're taking a pill. Hormones are really important, and there are these papers showing how not only female hormones, but testosterone, for example, modulates very, very strongly hERG blockade and hERG in general. So there are factors that are external to the cell that mean that our ionic currents are constantly modulated, not just by disease or mutations, but by factors we cannot control. So there is not a single conductance, but our conductances are being fluctuating all the time. And these are more factors. So we also looked at experiments quite a lot, and these are recordings of the human action potential. They come from the data that actually Yoram and Tomohara used to build the O'Hara-Moody model. And these are recordings from different preparations, and you can see the huge range of variability that there is in this human action potential. We think that this variability should come also from environmental factors like the ones I showed you before, in addition to other things like genetic differences. In this case, the human ventricular action potentials are measured in tissue, so this is not a matter of isolation. And they come from non-disease heart. So it could be that they are explained by a variety of factors, but it's not necessarily disease. So what we did is, and this is the simulation of the O'Hara-Woody cardiac action potential, which actually sits very nicely in the middle of a lot of our experimental data we have used. So what we did a few years ago is to go from a single action, well, the first thing is actually, before I mention anything, so this variability is actually consistent across species and across experimental types and across experimental preparations. So it's not a matter of experimental error in a certain experiment. We've collaborated with a lot of people worldwide and with a lot of different preparations and annual experiments, and we have reviewed the literature and we have modeling with this data, and we see that consistently there is a huge variability in the action potential measurements. It makes a lot of sense. If we look at each other, our noses, our hair, everything is different between each other. Why should the cardiac action potential be any different? Now, so what we did is to develop a methodology that would allow us to test certain hypotheses around what might explain this variability and the implications that this may have in explaining phenotype. So we went from simulating an action potential model, and it could be the O'Hara-Woody model or the Decker model or the Han-Woody model, to actually implementing cell-to-cell variability by using models that share the same equations, but initially where the conductances of the currents can vary very widely. The first assumption was that the variability that we see here is explained not by differences in kinetics, because we're talking about non-disease hearts, but by differences in the strength of the currents, of the conductances. And that was our first assumption. And so what we did is generate populations of models that included hundreds or thousands of models with the same equations but variation in the conductances. And we did that for a certain number of applications. This was the first paper. And what we do is we calibrate the models to include the ones that give us action potentials that are nicely in range with the experiments. So we have experimental ranges of biomarkers like APD or APD50, and we use the models we keep the models that are sitting within that experimental range. So this is an example. So this is the O'Hara-Woody model, and what one of the decisions to make is what are the parameters that we vary in the population and which ones we keep constant, because we don't have infinite computer power. And in most of the studies, what we vary is the conductances of the surface sarcolemma currents, but also in the calcium subsystem. And another decision to make, which is based on the hypothesis we need to test, is what is the range of that variation. Initially, we started quite small, and we thought that a 30% variation around the baseline was reasonable. But in fact, later, we went much more wild. And we said, why don't we make it vary more? We don't have any information on what is the range of variation in the ionic currents from the experiments just yet. 30% sound reasonable, but in fact, if we want to explore a lot of different possibilities, it makes sense to go small, like what if some of these currents are really, really small, like imitations, and how if they can increase quite a lot. So we went from 0 to 200% of baseline. And following calibration, so what we saw in most of the cases is that, sorry, I'm going mad with this thing. Okay. What we saw is that when we take into account a population of models that all have a normal looking action potential, these are non-disease action potentials, the range of variation of most of the conductances can be huge, can be really, really wide. For the sodium current, for example, this is not true. So we need to have a minimum amount of sodium current for the action potential to develop. And this is because there is almost a one-to-one relationship with the upstroke of the action potential. But for repolarization, there are so many currents that if one is downregulated, the others get counteract. So they balance, and you still have a good repolarization. In some cases, we could see some correlations, like between the sodium and the altered calcium current. And this is, in fact, also explained by Shaw and Woogie's papers, where if the sodium current gets downregulated, the altered calcium current can take over. But for the repolarization currents, for some of them, you can really have anything. you can have very low IKS, and that gets counteracted by high IKR and a lot of different mechanisms. And in fact, when you think about all the factors that affect these ionic currents, from environmental factors, it makes sense that a lot of these combinations should make an action potential viable. So, of course, what is this tool? So we build these populations and we have used them to understand different mechanisms or to identify likely mechanisms of arrhythmia. In this case, for example, we were investigating the response of action potential cells to Flecainide. This is what you see in the Hypertrophic model when you increase the dose of Flecainide. So if each of these curves is an increasing dose of Flecainide. In the population, what we see is that depending on the model, we see either a normal APD prolongation, depolarization failure in some cases, or EADs. So you have a different range of phenotypes that help us explain the variability in the response to a drug. And we've done that with several types of drugs, and this is in collaboration with some pharmaceutical companies who are interested in these methodologies. In this case, for example, this is the effect of Dofetilide on the population, and we see that some of the cells have a normal action potential for this drug of dose of Dofetilide, but some other cells develop EADs. The question is, what makes these two subpopulations different? Why should some cells develop EADs and not others? In this case, it's quite obvious that it would depend on repolarization reserve. So cells that have a weak IKS and a high altered calcium current would develop EADs much more easily than the ones that have a very strong repolarization reserve. In fact, one of the surprises is always the sodium-potassium pump. We always see that the sodium-potassium pump is much more important than we think for EADs development. And that actually is not even screened for in pharmaceutical companies. So we do get some interesting findings from this analysis. So the focus is really on, can we investigate differences in cardiac phenotype with populations of models, even if it is at the cardiac action potential level? And can we identify who and why might get an arrhythmia, in this case, at the action potential level? These are measurements that are conducted in London by Peter Taggart, Pierre Lamias, and Michele Orini, who some of you know, because he's been involved in ECGI. But in this case, what they were doing is recording using a sock of electrodes around the heart of patients that go for surgery, for other things, and they allow them to map the heart, and they get electrograms from the heart, 256 electrograms. We were interested in analyzing these electrograms for alternates, and we were interested in analyzing the alternates to see whether there were different types of alternates and whether we could explain the mechanisms of these alternates. So with the measurements, what we did is, first of all, most of you should be familiar with these cardiac alternates, and what we were looking in these electrograms is for patterns of short, long action potential duration, and in this you can plot based on cycle length. This would be alternates because you have short, long, and for longer cycle lengths, these alternates disappear. These alternates are not occurring at very fast pacing rates, but they are occurring at 400, 450 milliseconds, so they are not related to depolarization alternates. So the importance of this, and I think I should mention this because it's a seminal work by David Rosenbaum, who I know was a very good friend of Joran's. And he showed the importance of alternates in the survival due to arrhythmic risk. So these alternates may be a good indicator of proarrhythmia, and that's why the clinicians are interested in this. So there are two main hypotheses for these alternates generation. One is voltage-driven hypothesis, and this would depend on sodium, potassium, and calcium currents. And the other one is calcium-driven hypothesis, and that these alternates are related to fluctuations of intracellular calcium. So could we, building these our populations of models, explain some of these mechanisms and reproduce the in vivo phenotype that was measured by the clinicians? Again, we use the Hypertrophic model, and we vary the conductances of all the cerebellar currents as well as the release through the RYR and the SERCA uptake. And we vary these conductances quite widely in order to explore a lot of different possible scenarios. This is the population of models we built, and it's really nice because the O'Hara-Rudy model has nothing to do with this data. They weren't useful to build this model, but when we constructed the population, the O'Hara-Rudy model sits perfectly in the middle of this population and of this data. So this is all the tens of thousands of models we considered, and we only keep the red ones, which are nicely in the range of the biomarkers measured in vivo. And the O'Hara-Rudy agreed with the model, agreed with this data. So when we analyze the data, what we saw is that in vivo, in these electrograms, we could identify two types of alternance. One was a Y type. When we increased the pacing frequency, the alternance disappeared. And the other one was a fork type alternance. When the frequency increased, the alternance kept going. Interestingly, in the population, we saw models that never developed alternance, models that developed this type of alternance, and models that developed this fork type. So not only the O'Hara-Rudy model was able to reproduce an action potential, a population of models in range with the data, but also it was able to reproduce the different alternance mechanisms, alternance phenotypes, and we were able to identify that. Then what we did is to try to investigate what were the ionic mechanisms underlying these different types of alternance, and we used the population for that. And you can see that both RYR was increased in the models that had alternance in both of them, and SERCA was downregulated. And in fact, it was the loss of balance between the uptake and the release of calcium that generated these calcium alternance that were translated to voltage alternance through NCX. So we analyzed this population of models, and we were able to identify what were the key ionic factors that determine these differences in phenotype. In fact, what we saw is that the altered calcium current was higher in the I-type alternance. And because the altered calcium current was higher, the calcium builds faster, and that helps stabilizing the calcium alternance and leads to the I-type phenotype. All this is published in this paper by Shin. And what is more interesting also is that we were able to use the population to investigate antiarrhythmic therapy. So what was the strategy to decrease this alternance? And in this case, we tested NCX, and we looked at how blocking NCX would decrease the amount of alternance, and the population helps with that. So another study was related to hypertrophic cardiomyopathy, and this is an ongoing study because in Oxford there are people who are really interested in this disease. So this is Hugh Watkins is leading a lot of this research in cardiovascular medicine. So it's a genetic disease which is characterized by the thickening of the left ventricle. It's a very heterogeneous phenotype, and ECG abnormalities have been reported, but there is no consistent phenotype. And then there are also lots of cardiac magnetic resonance studies showing structural abnormalities. But the phenotype is very heterogeneous, and there is no real clear relationship with risk. So the first part of the study is to identify and characterize differences in phenotype in this HTM cohort using the clinical ECG. So this is clinical ECG analysis. And the second is to investigate mechanisms of repolarization abnormalities using computer modeling in hypertrophic cardiomyopathy. So this is a clinical study that is being conducted by Rina Arida in Hugh Watkins' group. And we have Holter recordings for 84 ACM patients, 39 healthy volunteers, but also blind to the ECG analysis, we have cardiac MR, genotype data, and standard clinical data. What we want to understand is, among these 84 HCM patients, do we have different phenotypes? And if there are different phenotypes, how related they are to risk, and how can we explain this risk? So this is the picture of the methodology. So we analyzed not only the standard ECG biomarkers, but what we did is we extracted the morphology of the QRS. So we wanted to go beyond just QRS width. We wanted to quantify what are the differences in the morphology of the QRS, and also T-wave biomarkers, and especially T-wave inversion. And then we use machine learning for clustering, and we identify different clusters of these patients based on mostly on the morphology of the QRS. And then we look for association with clinical risk markers for sudden cardiac death. So this is after a few years of analysis, by looking at different methodologies and stuff, we identified, based on the QRS, we identified 3 groups of patients. The ones that have a normal QRS, and these were more or less half of the patients had a completely normal QRS in all leads. The second group had an abnormal RS pattern, especially in lead 3. And the third one had a much more abnormal QRS. Here Watkins likes to call these fragmented, and the electrophysiologists don't like to call it fragmented. But just to give you an idea, what we quantify is how wavy the QRS morphology is. And interestingly, we could separate this group of normal QRS between those that had a normal T-wave and the ones that had a T-wave inverted. The ones that had a normal QRS and T-wave inverted were the ones that were at higher arrhythmic risk. So they also show differences in where the hypertrophy is located. But so this group 1A that has a completely normal QRS and a T-wave inversion shows higher risk score and higher risk of non-sustained ventricular tachycardia. So we are trying to investigate what makes that QRS normal based on the cardiac MR, and how these T-wave abnormalities can be generated in those patients. So we are interested in understanding why abnormalities in vocalization occur in hypertrophic cardiomyopathy patients. And this is a modeling study that was led by Alfonso Buenorobio, who works in the same department as me, and where we went back to the population of models to understand hypertrophic cardiomyopathy. So in this case, we used data by Elisabetta Cervai. So she has data that she has collected in human ventricular cells of patients with hypertrophic cardiomyopathy, and we built a population of models based on her data for control and for HCM. And we saw that the HCM cells were more prone to repolarization abnormalities. We actually recover, these are the experiments, and the values of the experiments, the cells in the experiments, and these are the simulations, and we were able to recover the phenotype in the cells. So what Elisa did was to, first of all, analyze the different types of EADs and repolarization abnormalities that we see in the population. And we saw that a lot of these different cells would result in EADs that were increasing in intensity. And this was very similar to what happens in the experiment. Some cells would develop single EADs, some multiple EADs, and some of them repolarization failure. And in the simulations, we were able to replicate those findings. So the next question was, what makes these cells different, and what are the main mechanisms underlying this? Because that might explain why some patients are at risk of suffering arrhythmias and some others don't. So I would think that so if somebody gets this cardiac phenotype, they would get more risk than if somebody has simple EADs or not EADs at all. So we want to understand what explains those different phenotypes. And what Elisa did is, again, to go to the currents and look at what makes the different cells different at the ionic level, identifying, obviously IKR is always very important, but also other currents are very important in balancing the currents during repolarization. So we were able to replicate the phenotype that is observed in the experiments and generating hypotheses on what are the mechanisms underlying these differences. And then what we did also, which is testable in the experiments, was to go and so we cannot easily test hypotheses on what we identify here, because this cannot be measured in the experiment. But what ELISA did is simulate the effect of drugs on the cells and on the ability of the drugs to suppress repolarization abnormalities. And we saw that NCOX block was very effective, complemented with a late sodium current or L-type current, calcium current block in suppressing these repolarization abnormalities. So of course, the next step is to ask Elisabetta to do the experiments where they generate these repolarization abnormalities and they block the NCX and look at whether that is actually effective. But our simulations identify these as a potential therapy. So, of course, what I have shown is mostly, except for the clinical study, is on isolated cells. And it's all very well to investigate early after depolarization in isolated cells. We are able to replicate these studies. But when we couple cells in tissue, and these are studies in a rabbit ventricle, I very, very rarely see any EADs. It's very difficult to generate EADs in tissue, and we were talking about that in some of the meetings I had. So the gut junctional coupling makes everything go very normal, very stable. So I've always had this problem with EADs, and other people share my problems with EADs, that I think it's a biomarker of repolarization reserve, but the causality with arrhythmia is quite unclear. So what is the relationship between EADs and arrhythmias? Actually, Andras Varos tells me that he's never seen EADs in his preparations in tissue, even when they block her. So is this a biomarker, or is there a causal relationship between EADs and arrhythmia? So I encountered this by chance, actually. So we were running this study where we were interested in understanding the effect of class 3 antiarrhythmic drugs in acute ischemia. Class 3 antiarrhythmic drugs block the potassium channels, and therefore they prolong refractoriness, and they're mostly antiarrhythmics, and that's how they are used. However, in clinical trials, they didn't work, and they increased mortality in patients with coronary heart disease in the SOAR trial and stuff. So we want to understand why this happens. We don't know. So we constructed a model of acute ischemia in the human ventricles. This is a model that Sarah Duta and Anna Michelet produced with all the knowledge we have on how the extracellular potassium accumulates, KATP activation gets activated, how acidosis affects sodium and calcium channels, and also heterogeneity in the endocardial and lateral border zone, as shown by a lot of people, mostly in animal models, but some information on human studies. So we constructed this model, and we simulated reentry. So we simulated the effect of an ectopic beat that gets blocked in the ischemic region due to post-repolarization refractoriness. The waves move around and re-enter through the ischemic region very nicely, like Janssen et al. Shown in pigs and dogs in the 80s. So the model reproduces this re-entry. I have actually a movie which shows this with the O'Hara-Woody model. So we managed to get the O'Hara-Woody model work in the whole ventricles, display post-repolarization refractoriness, and you can see here how the wave does what it should be doing, which is get blocked and then re-enter. We did these simulations in acute ischemia with hurt block. So we wanted to investigate how hurt block would modulate the vulnerability to re-entry in this model. And what we saw is that if we blocked hurt by 30%, the vulnerability to arrhythmia decreases because of APD prolongation. But if we block it further, we decrease repolarization reserve further by blocking hurt to 50%, vulnerability of reentry increases again, in spite of the fact that APD gets prolonged. So we had no idea why, but we looked in the simulations and we saw a very interesting pattern of propagation. What we saw is that the waves get blocked, and then there is a propagation through the endocardium that gets through to the epicardium in something that in an experiment, in an optical mapping experiment, would look like focal activation, because there is no way you can see what's coming from the endocardium. But this ectopic activity, which is not ectopic, it's propagation through, looks like focal activity. And that allowed for the re-entry to continue. Now, what makes this possible? When a postdoc came and showed me this graph, and I was looking at it, and I was thinking, but these are EADs. So these are EADs, and it was the first time I see EADs in tissue. So usually all the EADs are seen in single cell. When we couple them in tissue, they are suppressed. But in this case, what we were seeing is that in this region here, the wave propagation was supported by EADs formation. And it's hard to see, and it was hard to see for us too. But these EADs happen because there is a very, very, very steep gradient of potentials in that region. And the cells that are activated provide a very low amplitude, but very stable current to the neighboring cells. So there is a very steep gradient of potentials, and you can see some of this here. It's in the paper, but you can see that the gradient, some of these cells are already repolarizing when some of the others are depolarizing. And that provides a very low amplitude current to the cells that is enough to kick them back and activate the calcium cell again. So these EADs are occurring because there is low repolarization reserve. So the calcium current can bring the voltage up again. And they are caused by electrotonic effects that are due to a very heterogeneous substrate. So EADs occurred in this case not because of a spontaneous reactivation of the calcium current, but because there was an electrotonic flow of current that was enough to allow the calcium current to activate again. It makes a lot of sense because the arrhythmias usually happen in people who have an arrhythmic substrate, some sort of dispersion of repolarization. So it was actually only by looking at these graphs that we understood what is happening here. And that actually explains, or could explain, why class 3 antiarrhythmics don't work in people who have an arrhythmic substrate caused by ischemia, for example. We investigated the mechanisms of this, and you can see it clear here, the L-type calcium activation. And these EADs are occurring because of very low, sustained low amplitude current that is really, really small. It's not enough to trigger an action potential, but it's enough to destabilize repolarization and get an EAD. So this is things we have published. What are we doing now? So, I mean, we are doing a lot of the studies on cardiac cells and stuff, but I'm interested in going beyond that and understanding variability in the human whole ventricles, from the ion channel to the body surface potential. So I'm interested in understanding what makes the ECGs different, and what are the different factors that can come into play. And this is with a view of understanding variability at the whole organ level, which I think I'll spend my life doing it. So one of the studies is by Anna Mincholet, and what she's interested in doing at the moment is understanding the impact of anatomical variability on the electrocardiogram. So given a cardiac MR, we can reconstruct the human heart in a torso, so this is patient-specific anatomy, and we can construct a computer model of the heart embedded in the torso. Each of these steps is a person for many months, and there's a long list of authors here, but it's good because some of them are married, so they work very well together so far, and the others are friends, so they are happy to contribute to a big project like this. And then what we do is to run high-performance computed simulations where we keep all the factors that, like action potential model, heterogeneity, fiber orientation, everything the same in order to test the importance of variations in anatomy on the ECG. So this is run in a supercomputer in Edinburgh called Archer. So Anna so far has reconstructed the heart of these 5 people and some disease patients as well. So we're trying to understand also disease, but in this case it's normal people, and how variations in anatomy and also orientation of the heart affect the body surface potentials and the ECG in this case. So This is one good example. So actually to get a realistic ECG and a realistic QRS is not easy. And another student in my lab spent some years of his life trying to recover the activation sequence that leads to a good QRS complex in all leads. This is the best example, of course. So in this patient in particular, without touching anything, only with the anatomy, we could recover an ECG that is quite similar to the patient's one. In other cases, it's different, and it must be a lot of other variables are changing. In this case, what Anna is computing is just how the anatomy would make the QRS different. And in this case, for example, you can see that there is a relationship between the orientation of the heart and the QRS area. But she's going to do more of these patients. And the idea is to build populations of whole ventricular models. It's still basic science, so we're not trying to do clinical things like Natalia, for example. We're trying to understand different factors that may explain variability in the ECG in normal people, and then move to disease conditions as well. So just to conclude, we are interested in understanding different cardiac phenotypes, and we've done a few studies on experimental calibrated population, explain how much ability in Ionik conductances can explain differences in phenotypes. We have also explored some kinetics, and these are studies that are quite interesting. But we wanted to see how far we can go only with variability in INaCas. In the study by Shin, we identified two types of alternants that are identified in vivo, and the simulations are able to replicate it. Of course, this is a lot down to the Hypertrophic model that has a lot of the components in there. And the altered calcium current is the key to explain those two phenotypes. So in clinical study, we identified 4 distinct phenotypes in hypertrophic cardiomyopathy, and those with a normal QRS and inverted T-waves were at higher arrhythmic risk. In hypertrophic cardiomyopathy, at single cell level, we see that repolarization abnormalities may be counteracted by NCX and late sodium block, and I would be very interested in seeing those experiments done. So if we get the funding by Alfonso, we might see that happening. And gap junctional coupling suppresses EADs formation in whole ventricular simulations, but when there is a heterogeneous substrate, and we show this in acute myocardial ischemia, electrotonic interactions might facilitate the occurrence of EADs. At least this is what we see in the experiments. And we are interested in moving towards understanding variability at the whole heart level. It's an ambitious goal, and we're starting small with populations of anatomical models, and we hope to link this to ECGI to have a more characterized substrate. So that's it from me now, and thank you very much for your attention.
Thank you, Blanca. I was here before. Go ahead. I'll ask after.
So I was curious, the heterogeneous substrate in acute MI, do you think that part of that heterogeneity is due to the decrease in gap junction coupling that's happening?
Right, so that's actually a very interesting question. So that we haven't, in the model, we don't have that because we are simulating the first 10 minutes of ischemia. But in fact, if you have a decreased gap junctional coupling in that area, you will have probably more possibilities of EADs or less, I don't know. We should check in the model, because if you have less gap junctional coupling, it may be that you get less current through that triggers the EAD. On the other hand, you get less suppression due to the electrotonic flow by other. So it's an interesting question. This would be related to the phase 1B of ischemia. And it's something that we haven't investigated because you also have changes in calcium. So we haven't included that in the model, but it may very well be the case.
So on the same topic for the EAD formation, so it's really related to the source that you have and the loading on that source. So you can increase the source by increasing the number of cells that produce EADs in a synchronous fashion. And if you have enough of them and you have a critical mass, then even with large loading, they'll be successful in producing excitation. On the other side of things, the general principle is that you have to reduce the loading, and you can reduce the loading by structural things like fibrosis and gap junction uncoupling. That's one. But the other one that you showed an example of is by having an excitation pattern or repolarization pattern that functionally unloads a group of cells that produce an EAD. And there are many examples how you can do it. One of them is steep dispersion, the other one is a wavefront that comes and load the region that produces an EAD, so there is no loading. And this is exactly why Andres Varro, in any kind of tissue, you can produce those conditions. But Andres Varro, in his tissue, he stimulates from a point or he stimulates from a line. And under those conditions, he'll never see EADs in well-coupled tissue. So it's not surprising. But that's why not all of us have arrhythmias, because the substrate has to be pretty precise in order to produce those conditions, and probabilistically it's a very low probability in people who don't have structurally heart disease or another type of heart disease. The other question, this was in rational, but the other question is that
I just saw it and I thought, the first time I see EADs in tissue, and of course this makes sense, but we hadn't seen it before.
But I think who was, well, Crystal Ricklinger.
She saw EADs. Was that in the
I don't remember her study, but you remember that she talked about the minimum number of cells that you need. She has a number, and then Zilin Ku from UCLA computed a number, and they're pretty consistent.
So that was with the beta-adrenergic stimulation, with the neurons. Yes, but so what I see in the simulations and is that in order to get that, so also Sasha Pamphilov has Nele something, which is Dutch, I don't remember, Van der City or something, also show that you can generate EADs in ventricular models. Now, you need to have very extreme conditions. You need to increase calcium and decrease potassium and do all sorts of things. So it's not like a normal, you need to do maybe beta-adrenergic stimulation, but under normal conditions, it's hard, so you need to challenge the models a lot. Whereas here, we didn't do anything.
But normal hearts don't produce EADs.
But I think experimentally they were pushing them. So if you have a heterogeneous substrate, you don't need to push it so hard. That's I guess what we try to say.
Okay. But the principle is pretty clear. But I had a question in addition to this, which is, one strategy that you show that came out of simulation is to block the sodium-calcium exchanger. But if you block it chronically for a long enough time, you'll start to generate condition of calcium overload, and you might prevent EADs, but you will generate DADs, and you didn't gain much.
It could be. It would be interesting to see. So the Andras Stott, the guy we were talking about yesterday, has done experiments on the blocking NCX in reperfusion. It may be that you need, this is speculative, but it would be nice to see the experiments conducted, because it is true that it could generate EAD, but we don't know yet, especially in conditions like hypertrophic cardiomyopathy. I don't know. But in reperfusion, it's effective in dogs, in those experiments. So.
Did you see the EADs during the reentry? Is that what you were saying?
Yeah.
Did you ever see it if you just paced? You can't. So it's not an ectopic beat formation that can initiate reentry. It's actually occurring during the reentry.
In single cells, we didn't see EADs under those experiments. So in order to see, that's what I was trying to say before. So EADs in these models, if this was a Tertusian model, we're going to replicate everything with the O'Hara hoodie. You need to block the hair current a lot to see EADs in this model. And we were blocking it by 50%, so we weren't even seeing a huge increase in ADP. But the kick of this low amplitude current was enough to destabilize the APD and get the EAD. So it is possible that this heterogeneous substrate provides this low amplitude current that makes everything stabilized. Yes.
Did you look at whether there's an increased predisposition near the Purkinje myocyte junction?
We don't have Purkinje here, but that's a good question. We don't have Purkinje in this model, but it is possible. I mean, all of those are possibilities for acute ischemia. what we are proposing is yet another form in which these arrhythmias will be sustained when you have low revitalization of reserve, which wasn't considered before. And of course, you can add to that everything you want, but at least without all those factors, we can see that, which makes studying more interesting for me, at least.
Very interesting. I have two questions. One of them is that when you vary those ionic currents, are they correlated or related, or are they just completely independent?
See, I mean, that's a very interesting question. I mean, we were talking about that with Colin before, when we let them vary freely. We use a sampling method, which is Latin hypercube sampling. And you could sample them in any way you want. You could include correlations if you wanted, but we haven't included anything. The reason being that we don't have any experimental data backing correlation. So we looked for those. Statisticians love these correlations, so they ask for that because they have a bunch of methods that allow them to analyze it. But there is no experimental backing. The only paper we could find was on the sodium current and ICal, where they were showing that there was some relationship between the two. It's not really correlation. It's whether if you suppress, is that Khalifa's? Yeah. So when you block one, or the other one gets overexpressed, which is not really the same as having a correlation. So if you show me correlations, we can implement them. But in fact, we don't. So we let them run free. So it's kind of an exercise of freedom. So you let them run free. What do we see? Most of the cells would have a normal phenotype. The ones that don't have a normal phenotype, why?
So one of the things that I don't see. So could you have like a perfect storm that when all the potassium channels are lower? So I mean, what you don't see those kind of extremes.
Yeah. So we have depends on the model and depends on the study. You see different things, but most of the time we can see very wide profiles. Huh? So in some cases you do see a little bit of a correlation. Interestingly, in some of the drug studies, what we saw is that the abnormalities that happen when you block multi-channel, multiple channels because of drugs that are dirty and they were happening in cells that have a very, very low ICal, like so low that we thought these models cannot really exist. So Elisa Bassini again was Googling, for whether that can be possible. And in fact, there is a mutation that affects some families that live with very, very low ICal, and they have cardiac arrhythmias. So we believe that a lot of these phenotypes are possible in the human population, even though initially we are used to not thinking about that. It's a bit scary because it makes everything much more complicated.
But I come back to what we were talking about earlier, I think, which is that you have all this variability, and yet you don't have all that much variability if you actually look at the single cell action potential.
Well, I mean, you do see.
Which means that there must be a correlation somewhere.
Yeah, you missed the beginning of the talk,
sorry.
but no, it's not what I mean. I can show it to you. So this is the measurements of the human ventricular action potential in non-disease heart. So is that right that we don't have a lot of variability in the experiments, or that the papers don't show it? Because if you show a lot of variability in the experiment, what are your reviewers going to say? So, because initially, I mean, I had this experience of talking to experimental collaborators about variability, and they were like, "Oh, I don't want to touch that because my p-values are going to be a mess, and I don't want to show people that." And in fact, we tell them that everybody has the same type of data. It's okay. We are variable. We are different. You don't need to hide this variability. Let's explore it because it's related to medical conditions.
I can attest to that. That's totally true. Tons of variability.
But also that people don't want to show it because it looks bad in papers. But what are clinicians trying to do? Understanding variability. So maybe we need to,
Wouldn't some of those be
recordings? There's tremendous variability.
But some of those can be considered as longitude, right? I mean,
well, maybe, but
It's single cell, but once you put them in a heart, you want
in
to
the heart,
I don't know. It's a longitude is defined by the body surface ECG. So the fact that some cells will be very long and then in the tissue they will be loaded and
But in fact, so also, you know, we have a good relationship with all these people, so they know so we understand that, anyway, so experiments are hard and all of that. Now, so if you compare, so these data, for example, here are from Ursula Raven's trabecular, and the ones that there, human atria, so this is human as well, are from Barbara Casade's lab. So this is long, and that is short. They both come from human preparations. They are both excellent researchers. What is going on? Can we build models that try to embrace this variability? It starts to think, you know, to look like let's embrace difference and all of that. But it is true that when we review this, it is true that there is a lot of variability, and it makes a lot of sense.
But it says it's isolated, so I wonder if
Yeah.
when they add all these enzymes,
Yeah, but-
some of those
Yeah.
cells feel the enzymes a little bit
That
more.
one, yeah, that is
Yeah.
isolated. This
Actually,
is not isolated.
Yeah, there's more variability in the atrial trabecular,
Right.
which is the intact tissue.
And in fact, when you isolate cells, you're supposed to kill some potassium channels. So usually you will have the opposite effect. You have a prolonged variability. So why is that? But the fact is, we can build models of that preparation, and we did it for Barbara. And then we looked at she had the, well, we are part of it, but it's this paper on NOS inhibition in atrial fibrillation. And we were able to build models that, the same Courtemanche, Malekar, and Grandi models that are able to replicate these findings, were able to replicate that by changing the conductances only. And in fact, we were able to include the effects of NOS inhibition and reproduce the experiments as well. So it is likely that all of these differences are due to, in big part, conductances. Of course, kinetics, and there are studies coming out on variability in the kinetics as well. But conductances can vary a lot.
Well,
As to
also-
the ECG, I mean, the big problem is that the ECG is the source of the electrocardiogram, really, is the spatial Laplacian
Yeah.
of the transmembrane voltage. So it's not an easy relationship that you can say if the action potential is prolonged, one,
Yeah.
or a region, then you have prolonged QT interval on the ECG. It's not true. It's not a linear relation.
But those don't take into account also where in the ventricle and where in the wall, endo, epi, those kind of things.
Yeah,
right?
that's right.
So maybe it's not as, maybe there is some
So-
regionality to.
The human atria are from the same place because
Right.
that's when they.
Yeah, that's really markedly variable. How do you explain that?
Yeah. So it's the same so from the same location, because it's when they remove the, it's a clinical procedure when they get the tissue from the appendage, is that how you call it? And I mean, it is true that it could come from variability of different stages. We have no evidence that it can be only that. So I think, I mean, and this is a belief, it's like a religious belief. I think there is a lot of variability.
Yeah. You might be uncovering a very dirty secret.
It's actually fascinating.
Yeah.
I think it's really.
The ventricular model that you had a conduction system
No,
in the
no,
ventricular
no.
model?
We are simulating activation by, so not yet, and that because also we don't have a good human ventricular Purkinje model and all of that. So we don't have that. We are simulating the ECG by putting root nodes for the activation to propagate.
Sure.
So that's something we can control.
You generate some initial conditions.
Yeah. And I was hard enough actually to have a Purkinje.
It's not easy.
Yeah.
So your table shows variability between cells. Is it known how much the same cell will vary on a dynamic
Yeah,
basis?
so that's the bit-to-bit variability, for example. Is that what you're
Yeah.
referring to? So, yeah, so we have also studied that. I didn't speak about it, but there is variability bit to bit. And one of the hypotheses we investigated is whether that variability comes from the stochasticity in the ionic currents. So in fact, we did that for IKS. So and we show that the variability, the small variation in IKS can reproduce a lot of the variability, the bit-to-bit variability in APD. If we couple those cells, so in isolated cells, we are able to explain that bit-to-bit variability. If we couple them in tissue, we don't. So it must come to another
In
place.
isolated cells, there are not many of the key currents that show significant stochasticity.
We
they're
publish
bit-
it in Biophysical Journal. So we show that stochasticity in IKS, and we calculated numbers of channels, and we used a stochastic equation with the Lambda-Dean equation. Only the IKS contribution could explain a lot of the bit-to-bit variability. If you couple in tissue, it's gone.
What about KTP? Because KTP has a bit much
Yeah,
bigger.
I don't know.
Because the
It's an
current,
interesting
the single
question.
channels are bigger,
Yeah,
so
it could be.
but even then,
So bit-to-bit variability, and we have another study this year where we included stochasticity in other currents, and we show that we can replicate a lot of the experiments with a dog model.
so are you saying that IKS, the whole cell current, is noisy enough that, and the impedance is high enough at the right point of the action potential for it to really change it?
And there are some sets, I mean, it's not only us, but we also, John Hyman has looked into this and other people. Now, again, so you couple things in tissue and that small variation is gone. So people are looking at bit-to-bit variability as a biomarker of arrhythmic risk as well for drugs. So whether that actually tells you something about repolarization is there. But I don't think that's the reason why you get, variability in the ECG, for example. It needs to be a stronger mechanism.
Anybody else? Well, thank you again.
Yeah.